Thanks for the good introduction. My name is Kwangmoon Park. I'm a fifth-year statistics PhD student here. So today I'm going to talk about learning multi-way interaction from single-cell Hi-C data set. So this project was mainly motivated by this science paper that talks about this MINTsC gene, which is a key oncogene governing the cancer cell proliferation. And it is known by the literature that this MINTsC gene is mainly controlled by the 7 enhancers, which you know that's E1 through E7. 4 of them are the immediate downstream of that MINTsC gene, and the rest of them are quite far apart from the promoter region of that MINTsC gene. And the question that this science paper had is, what are the interaction among those enhancers in terms of regulating the expression level? And therefore, they design a guide RNA-seq that can knock out one of these enhancers either individually or as a pair. And what they found out was that if they knock out those enhancers from different regions, then there is some interaction effect among them in terms of controlling the expression level. What I mean by that is if you just only knock out the E3 from the blue region, then there's like a certain level of drop in expression level. And same for the E7. But if you drop all of them at the same time, which is at the last end, then the magnitude of the drop of expression is way larger than expected by the additive model. The expected region is in the purple line. And that it means that there are some interaction effect among these 2 regions, and which exist and important. What this brought us is that, well, the aesthetic SNP interactions are not much addressed in GWAS or EQTL studies, even if the sample sizes are growing these days. I mean, what I mean by that is, if we imagine a SNP in E3 and another SNP in E7, then there should be some SNP interaction effect in terms of controlling the gene expression level. And that we thought that is not really addressed. And maybe the reason is finding interaction effect in EQTL analysis would be kind of cursed by the dimensionality or multiple testing issue. We cannot really consider all the combination like that large with that sample size. And the key question that we had is there a way to generate a candidate of those multiple regions based on the physical interaction? And hopefully one region can contain the promoter region of a gene, and the other 2 regions could contain some enhancers of that gene. And typically when we consider the multi-way interaction among these multiple regions, we're talking about the case where there are more than 2 regions contained. And actually, in terms of physical interaction, there are technologies that can do that, but they provide some proof of concept with application to cell lines. And we think that this is still a boutique technology because for each technology, there's about like maybe 1 paper and like 2 or 1 or 2 data sets available. That's relatively compared to like Hi-C data or single-cell Hi-C data. So Hi-C is most widely used or produced data set in terms of this 3D organization. So to give you an overview of for this Hi-C, you can consider contact map at below, where each entry denotes the contact level between 2 locus pairs. The red region denotes there's more contact than the other. And now there are single-cell level sequencing for this Hi-C, and there are a lot of labs generating them, a lot of data sets available publicly. But the thing is, the single-cell Hi-C or Hi-C, or they are all pairwise. They're not giving us something about multi-way interaction among multiple genomic regions. So given this fact, our key question was, can we infer some multi-way interactions based on this pairwise single-cell Hi-C data? And that's what we do for this project. So to give you an insight of what we thought about, so let's say I ask you if there's a multi-way interaction among this pink, purple, and green one. And you observe from 1 single cell, all 3 pairwise interactions are there. Then you can, in the noise-free setting, in the ideal setting, if that is observed, then you can safely say this is multi-way interaction. But the reality is not like that. We have the single-cell Hi-C or Hi-C, they're all proximity ligation based, and the short-read sequencing may limit the number of pairwise interaction observed. Even if, in truth, there is a multi-way interaction, there could be some pairwise interaction that could be lost from the data. That's like a bad news. But as a statistician, there's also a good news. We can observe that across cells. So these can be considered as samples, and what we can actually infer something about what's happening at the population level in the true sense. So calling multi interaction becomes a statistical problem, doing something about population from the sample. So we actually first enumerated how many multi interactions or clicks are observed from the data. I looked at the fully connected clicks for either in low resolution data on the top, high resolution data on the bottom. And you see that there are a lot of like multi-way interactions observed, mostly three-way interaction, but it doesn't really reach to like more than 7 or 8. So based on this observation, if you can enumerate how many clicks, candidate clicks are there are, then the question now becomes, for a given candidate multi-way interaction or click, how can you tell if this is real or not? And that requires constructing some test statistic for each click. And as I said, single cell high-C or high-C are pairwise data set. So we need to come up with a null model that also first start from like locus pair level, pairwise level, and then build it up to the click level. That's what we are doing for this model. So essentially, we're saying that this giant contact map that's following multinomial distribution. And there's one thing to note here. So the contact probability for each of them. So note that here the sample unit are all locus pair of that contact map. And one thing to note is that the contact probability between locus pair depends on the genomic distance between the locus for that locus pair. So if you imagine two loci that are very close to each other in genomic distance, then it's likely that they're going to have higher contact. And based on investigations, they have like a decaying nonlinear pattern, so we need to incorporate that. But the thing is, if you try to fit like a GLM model on this raw data at the locus pair at single cell level, and imagine doing like a 10 base pair binning, then it's going to be several millions data points, which is going to be very slow. Then maybe we can use a trick that we know from probability courses. Like, so first of all, we denote this one off diagonal as one band, a set of locus pairs that are having same genomic distance. Then we can actually aggregate the contact of those locus pairs within a band into a single number by just simply summing it up. Then by the multinomial distribution aggregation property, you can have a smaller multinomial distribution, and you can approximate that by Poisson distribution. And okay, you can only fit that model with this band at this band level, which is going to be GLM spline, Poisson GLM spline. Then if you obtain that PJ, then you have some parameter estimated for each band. But what we really need is not the band level probability, but we need to do something about the locus pair, the original level that we are considering. Then if we have this PJ estimated, then we need to somehow distribute each PJ into locus pairs. And that's why we model this as with a prior distribution with Dirichlet prior, and the prior distribution can be estimated based on mutability or GC, or if there's another cell type information, then you can also use that to estimate that. And based on that, you can set a null distribution, and then based on the null distribution for each locus pair, you can build a expectation and variance simple calculation. Then note that you can derive a test statistic for each locus pair, which is the building block of the click level test statistic. And this behaves, if you construct a z-score, then it converges to normal distribution if the cell number increases, and the corresponding p-value for that follows normal distribution under the null. But as I mentioned, we want some statistic for the click. Then let's say you're considering this click, and we have several p-values there. And what is the one way to summarize this? Well, we first thought that we might not really need all the pairwise interaction to be super strong. Maybe we can control like R or the statistic of this p-value to be strong enough. For example, this three-way case, we can say that two of them are very strong. Then we can say that this could be multi-way interaction. This R can be chosen by the users. Anyway, for our MINTsC, we consider R, so the statistic of these multiple p-values pairwise. then under a given set of null, it is known that this R, so the statistic of these p-values follow beta distribution. And based on that, you can obtain p-value for each click now. Each dot here is actually a click, and this is telling you how likely that click is to be multi-interaction. Then with this p-value, what you can do is FGI control, Bonferroni, whatever you want. And you can infer something about multi-interaction based on single-cell Hi-C. So we did a lot of validations using some orthogonal data sets. What I'm going to show you here is one of the boutique technologies that I mentioned. One of them is Sprite. So it directly tells you if one click is likely to be a multi-interaction or not. And based on MINTsC, we can also do FGI control and based on p-value, and then say this is significant or not significant. And you can see that the Sprite score is higher in the case where you can tell if this click is significant. So there's like a agreement between these two. And we also took a look at the spatial distance, average spatial distance of each click. So for each click, you have load and you have the spatial information from DNA-seq fish. Then you can just simply calculate the average distance among them. And what you would expect is that if it's less likely to be multi-interaction, then the spatial distance should be further apart. And that's the trend that we see here. The signal looks kind of weak, so I permuted the data with some structure, and the trend is not anymore there. So this part is actually what I really wanted to talk about. So based on MINTsC, we can say one click is significant or not. And so this was the data application for human prefrontal cortex. We took a look at one specific click with very high signal, and that region, that multi-interaction contained one locus containing the DKK3 promoter, and other two loci contain the enhancer with epigenomic signal in that specific click. Then we considered all the SNPs residing in these locus pairs, and then we fit multiple interaction EQTL model based on the Ross-Met Alzheimer's disease study. And the corresponding p-value for this guy, the interaction effect, is plotted on the QQ plot. And the gray ones are obtained based on the permuted data. And you can see that the signal is there for the interaction effect. And it chose a specific interaction effect model that contains two SNPs and one gene. And what you see here is that if there are only one minor allele, or both of them without minor allele, then the expression level is not that much changing. But if you have like both of them minor allele at the bottom, at the very right, you see that the expression level is higher than the other cases, which is showing us that there's some interaction effect on these SNPs for the expression level of this DKK3 gene. Okay, so this is what I had, and I want to thank my advisor, Sundus Keles, and Sichi Shen from our group. He helped me a lot. And I just got revision requests yesterday, and I should go through that one. But I also want to thank my collaborators here. Thanks.
And we do have time for questions.
So do you think MINTsC is finding physical interactions, or is it finding statistical interactions?
Sorry?
Is it finding physical interactions, or is it finding statistical interactions?
It's finding physical interaction. Yeah, and try to do.
But you're using pairwise data.
Yeah. So the interaction for physical contact and the interaction, all those interaction, the terminal is like, sorry for the confusion. It's about the physical interaction.
I was wondering if you could maybe give advice in applying the method because it's being used to constrain regions for, I guess, EQTL or a GWAS study or something like that, but you need to use Hi-C data to get the prior regions. So the Hi-C data will come from some cell type, but I guess it caught my attention you're using was AD RNA-seq data in this EQTL study. So do you have thoughts about what sort of Hi-C data you can use across different EQTL studies, or just generally how you map across maybe expression data and high-C data, because that seems to be a perennial challenge.
Yeah. So for this case, Alzheimer's disease, we typically look at only like brain tissue, and there are data sets for brain tissues as well.
So you had high-C data for brain, okay.
Yeah. But people could use just aggregation of like single cell high-C met and take a look at the regions that have high contact. But that still has noise. Like just taking average might not be like optimal. So.
Okay, thanks.